So today we are doing part 1 of the biological macromolecules lab, and we're going to do part 2 on Wednesday. And when I say we're going to do, I mean I'm just going to give you a run through of it. And then if you want to do it with me today, that's cool. I'll be here on Zoom. You can even sit through the next session if you would like to. I'll be here for a few hours. We're going to start out with questions, and then I'm going to review the biological macromolecules, which I think is something that some of us might need. And then I'm going to give you a quick run through of the lab, and then we're going to talk about what's on the quiz on Wednesday. So the only new assay that we're running this week is using Sudan3 to check for the presence of lipids. Everything else should be relatively familiar to you. It should be things that you've seen before, including the topic. We've talked about biological macromolecules since early on in the semester, so I just want to make sure that that's something that's solidified in all of our minds. It's something that we're all reasonably familiar with. So before we get started, are there any questions that anyone has about stuff? Format.
I have a question.
Yeah, go ahead.
So I was looking at stuff that was due. I ended up doing my stuff last week just to get ahead. And on the if you look at your grades, it says a bunch of stuff for week 9 discussion, quiz, and exam, all stuff that says it's due on the 13th of March, but there's nothing in the modules. So I was just wanting to ask about that.
Sure. And that's probably just an artifact of me pushing things back a week, which I did last week.
Okay.
The things that are due the 13th are going to be your mitosis. Wait, or was that one already due? What's definitely going to be due is going to be the biological macromolecules.
Okay. This
week's lab. Let me look here and make sure I know what I'm talking about. There's a quiz.
Mm-hmm.
There is the what are you curious about discussion. And then there's the biological macromolecules.
Okay.
That's all I see. Let me check the calendar just to triple check and be sure that's exactly what it says.
Okay.
And then let me click here really quick.
I just wanted to make sure because I was just a little confused about that. And then
no, that's great.
No,
that's a great question. And as you are probably discovering, I'm not the best at using the calendar myself. I kind of just go by the modules, and sometimes things get squishy with that.
That's okay. I just sometimes will look at my grades and stuff, and then I just noticed that it had week 9, and then where it says photosynthesis is due on the 20th. So that's why I was a little confused. So I just wanted to make sure.
Yeah, exam 2 is not due on Sunday. So I'm gonna have to, right after this, I'm gonna have to go through and fix all that stuff up, because I pushed things back, but apparently I didn't push everything back. So I will work on that. So what's due this Sunday, though, to be clear, is the biological macromolecules lab. What are you curious about? And the quiz, the week 8 quiz, I believe.
Okay, thank you.
Yeah, sorry about that. That one's on me. I don't operate using the calendar, and obviously I need to start doing that. But yes, I will fix that for you. My apologies. Anything else? Okay, well, if you have questions, feel free to stick them in the chat or wait until after we're done talking today, and I will get those answered for you.
I have one really quick. Do any of the labs span over a period of longer than a day or 2 days?
Any of the future labs? Yes.
Okay.
Yes, they will. Why don't you stick around after, and we can talk about what your concerns with that are, and make sure that we get that shored up and we're on the same page moving forward.
Okay, sounds good.
Because yes, there will be labs that we're going to be growing some seeds and doing some different things. So yes. Anybody else? Okay. Let's go ahead and trudge forward. So let's talk about the 4 biological macromolecules. Again, this should be review from way back in the day, but it's important to me that you know what the structure and functions of these things are, okay? So lipids. Lipids are these long chains of carbon. Okay, they look something like this. This is like a triglyceride thing. But you can see there's just these long chains of carbon. Maybe sometimes you have some differences, like there's a double bond here, or like maybe you'll have like this nitro group, or I think that's nitro. You have a nitrogen group here. But you should know that these lipids are long chains of carbon. Is probably also useful for you to know that these are not soluble in water. Okay, so oil is an example of a lipid. Oil does not dissolve in water. And as I referenced way back in, I think week 2, everywhere where you have one of these points, there is a carbon atom and all of the hydrogens that can fit with it. Okay, so this might just look like a bunch of random lines, but this is in fact carbon, 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 carbon. It's a long chain of carbon. Okay, and that is what defines lipids. Lipids are the primary constituent of our cell membranes. Remember, these are phospholipids. So you have this phosphate group up here, and then long chains of carbon coming off of that. Okay, so lipids make up our cell membranes. They are also used for the long-term storage of energy. And so if your body has a whole bunch of energy, it's going to make what's called glycogen, and ultimately it's going to be stored in the form of adipose tissue in your body. Okay, so lipids can be used for long-term energy storage, and they're principally used as your cell membranes. They can also be used for cellular messaging. Okay, so your body needs to send a message from one cell to another, or even within a cell. Lipids can be used for that. Oh, sorry, I will enable that. Cool. So yeah, lipids, long chains of carbons, that's how you identify them. They are your cell membranes. They are used to store energy, and they can be used for cellular messaging. And to be honest with you, the cellular messaging stuff is a little bit beyond the scope of this course, but it has to do with like sphingolipids and G protein activation and all kinds of, I think, really interesting stuff. But just know cellular messaging, nothing beyond that. We also have proteins. Proteins is another one of our biological macromolecules or biomolecules. These are chains of amino acids. It looks something like this. And you might look at this and say, okay, well, there's a carbon and there's a carbon and there's a carbon. Is this not just another lipid? And the answer is no, this is not just another lipid, because it follows a different pattern. It's like carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen. And what really makes proteins special or unique from one another is that they have these R groups, okay? And these R groups are what make up the different amino acids. And kind of what I'm getting at here is you can think of proteins maybe kind of like Legos. You can put different pieces of Legos together to make a larger structure, okay? And each individual Lego piece is going to contribute something different to the structure. Maybe that's a crappy metaphor, I'm not sure. But these are made of chains of amino acids. They also have a great deal of structure to them. Okay, and you don't need to worry about primary, secondary, tertiary, or quaternary structure, but I think it's important that you hear those terms. And I also think it's important that you know that these things are fairly intricate. Okay, and so when you do things like we've been doing, like boil them, the enzymes cease to function. They get too hot and their structure comes apart. These are very often, as we've seen, remember those transmembrane proteins. These are seen in your cell membranes as well. Proteins also function as enzymes, which hopefully we all know a little bit about. Those molecular catalysts that make reactions possible by lowering the amount of energy that you need to start them. The next category is nucleic acids, and hopefully remember that it is a phosphate sugar backbone with nitrogen bases. So I don't know if you've seen this particular picture before, but phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And that's just that pattern is gonna just keep on going throughout the entire molecule. And then you'll have nitrogen bases in the middle here, okay, connecting the rungs of this double helix. DNA is used for storing genetic information. Deoxyribonucleic acid is used for storing genetic information, and ribonucleic acid is used for transmitting that information, or for actually turning that DNA into a protein. Okay. Ribonucleic acids are also sugar phosphate, okay, which you can see here. Sugar, phosphate, sugar, phosphate. It's depicted a little bit differently between these two pictures, but it's showing you the same thing. But RNA is a single helix, whereas DNA is a double helix. And we're going to get into more of the RNA stuff in the coming weeks, and we're going to learn all about how that DNA is actually the recipe book for all of the protein in your body. The next one is carbohydrates. The last one is carbohydrates. These are rings of carbon, hydrogen, and oxygen. Okay, and here's one that hopefully you should be familiar with. Here is lactose, which is a glucose and a galactose. Here is a molecule of glucose. And these are used for quick energy for the cell. Okay, just fast. You know, you eat something and your body can quickly convert it into ATP. You also will see these things, even though I've told you they're rings, sometimes you'll see them like this in this form. I don't think I ever intentionally show you something like this. I'm always going to show it like a ring, but maybe you can see that this picture and this picture are actually the same thing. One's just kind of drawn end on, I should say, so that you can see it a little bit better or a little bit differently. Okay, these are for energy storage, but you also see carbohydrates within your nucleic acids. So, you know, remember, it's phosphate, sugar, phosphate, sugar. You have a bunch of sugar inside of your nucleic acids, okay? So should also probably give you a bit of a sense of scale for these two things. Nucleic acids are much larger than sugars. Okay, so part one of biological macromolecules, you're going to need to boil a little bit of water for this lab, so do be careful. A bottle of distilled water, or again, you know, as long as you can drink your tap water, it should be fine for this. Things that you might need to buy are, you need an egg, and you will need you don't even need a quarter cup, but I don't know if you can buy them any smaller than that. If you don't live in a milk drinking household, get one of the tiny little carton thingies. And a little bit of vegetable oil. I think vegetable oil we're going to be using for the second part of this exercise. But if you do decide to do this, I would recommend trying to do it in one sitting, because you're going to use some of the albumin from the egg, or you could just crack open another egg. That's fine too. You also need a small potato for this. It says russet, which just get a russet. I'm imagining you could probably use pretty much whatever kind of potato, but russets have a lot of starch, and that's one of the things we're going to be actually looking for again today.
Is
Sorry,
is a regular spoon okay instead of a plastic one?
It's for mixing, so you should probably use one that you can dispose of, but I think it'll be just fine. Yeah, I think it'll be fine.
Okay.
Because I don't think we're mixing up the biurettes with it or anything like that. But we'll find out in just a minute. I'm trying to think when I did this lab the other day. I'll say a regular spoon is probably fine, and we'll find out when we go through it.
It's
Okay.
used for
Sounds
testing the potato with the IKI indicator.
Pardon me?
It's the spoon is used when you're testing the russet potato with the IKIs.
That's right. You dig out a little chunk of it. Yeah, it should be fine. Just make sure that you really clean your spoon very, very well.
Will do.
And yeah, and make sure it's a spoon that you don't necessarily care about in case you do need to chuck it. But I don't think iodine's gonna stick on the spoon or anything like that. Okay, anybody else? Are we all good? Okay, hopefully my GIF works here. Oh, sweet. Okay. So step one is you do need to put on your personal protective equipment. I'm not doing the lab right now, so I don't have any, but please put on your goggles, please put on some gloves, which is going to feel silly for at least part of this because the first thing you do is crack open an egg. So you crack open your egg, and then I don't know if you guys have ever baked or made like a yolk-free omelet or anything like that, but you basically pass the yolk back and forth between the two pieces of the shell so that you can separate out the white. Okay, the white part is what we want. So then you're going to go ahead and grab one of the jumbo graduated pipettes. It's just going to be one of those long ones that have the graduated marks on the side, and you're going to label it albumin and put it with your, sorry for going too fast, put it with your cup of egg whites that we just gathered. Then you're going to whisk the egg whites up. Then we're going to label our test tubes DH2O, again, tap water is probably fine, albumin, milk, glucose, and egg white albumin stock. And we're going to put those into that 24 well plate just to use that as a temporary holder for right now. Ooh, some folks have broken test tubes. I hadn't considered this. If you have a broken test tube, I might suggest using another one of the cups instead of the egg white albumin stock test tube here. Yeah, I think that's what I would say. Because you don't really, we're not going to use it for any reactions or anything like that. Again, I think ideally the best way to do it is to have all the test tubes, but if one of them broke, which totally happens, maybe just do the albumin stock in a separate, in another one of your clean plastic cups. So the next thing you're going to do is we have that cup with our egg white. We're going to take our albumin graduated pipette and we're going to transfer about half of a milliliter of egg white into that stock test tube. And again, if you don't have all of the test tubes, it's probably okay to use a separate cup. The best thing would be to have all of them, but not everyone does. So you'll also notice there's graduations or there's markings on the side of each of those pipettes. So you can use those to estimate about a half of an ml of your egg whites. This is not perfect. This is not as good as the graduated cylinder, but an estimate here is just fine. Okay, so I've added my half a ml of albumin to there. The next thing that you're going to do is you're going to measure out 4.5 mLs using your graduated cylinder. You're going to measure 4.5 milliliters of water and put that into your stock cup. Gently mix it up, gently stir it, swirl it to mix. So there you go. We added our 4.5 mLs of water right there. Then you draw from that stock using your graduated pipette to add 1 ml of your stock to just your albumin tube. So again, let me go back. What's happening here is you have, I went way too far. You have 5 tubes. You have an albumin tube and you have an albumin stock tube. And that's so that you can run multiple assays using just the albumin tube. So you can draw from the stock and you don't need to, but you'll see what's going to happen with the albumin tube. Okay, we've measured our 0.5 ml and our 4.5 ml into our albumin container. Then we've grabbed our 1 ml of egg white albumin and water solution. The next thing you're going to do is just add a milliliter of water to your DI tube, and then you're going to add 1 ml of milk to your milk tube. Okay. Then you're going to add 24 drops of glucose. Remember your little glucose dropper bottle. You're going to add 24 drops into your glucose tube. Okay, so then you observe each of the colors of each of the samples and record those in data table 1. Then we need to boil some water because we're using biurettes, and maybe you remember biurettes from weeks ago. It needs to be heated up in order for the reactions to take place. Anybody remember what biurettes looks for, by the way? Okay. Maybe check out, you might need to check out the lab to figure some of these things out, but that's something I would know. So we've got our boiled water, and you're going to fill the 250 milliliter beaker about halfway full. Please use an oven mitt. Please be careful. Please also be wearing your regular gloves and your safety glasses. So the next thing you're going to do is add 40 drops of benedicts reagent to each of the 4 test tubes. Okay, and so not the stock test tube. So we're only running a test on water, albumin, milk, and glucose, not the stock albumin tube. Okay, hopefully that's reasonably clear. And if it's not, just get another egg. So after we've added our benedicts reagent, you are going to put your 4 test tubes into the hot water. It needs to be above 80 Celsius, I believe, which is quite hot, too hot to touch with your bare hands. So water still needs to be quite hot, and you need to let it sit for 10 minutes or so. Take the test tubes out of the water, put them back into that 24 well plate, and observe the color of the 4 samples and then record the data in data table 1. And then there is a figure inside of the lab that shows you an approximate quantity of reducing sugar, and that is on the lab. I will try to remember to stick a picture of that onto Canvas just in case it isn't loading for people, but I don't think it's a problem. If you can't see figure 11, please just shoot me an email and I will send it to you in case I forget. When all the data has been recorded, you can go ahead and pour those things down the sink. That biuret is pretty nasty, so just make sure that it gets flushed down with plenty of water, and then wash those 4 test tubes out, not the albumin stock one, because we are going to use that for the next part. Okay, so you're going to use the same 4 test tubes, except for you're going to label one starch this time. Which one did we replace? So dumb. Oh yeah. So instead of glucose, we're going to be testing starch this time. Use the albumin pipette to get 1 ml of albumin and put it from your stock into the albumin tube. Hopefully it's clear why we did this, because we're using albumin for more than one thing, and we don't want to have to remake it every single time. Okay, so we've got our albumin transferred over. Then we're going to add a ml of water to the water tube, and we're going to add a ml of milk to the milk tube, and then we're going to add 24 drops of starch from the dropper bottle to the starch test tube. So in the previous exercise, or not the previous exercise, in the part we just did, you add glucose from your glucose bottle. In this one, we're going to add starch from our starch bottle. Let's see if we have a question here. Okay, somebody asked if there are only 2 exercises in this lab, and kind of yes. There's a part 1 that has like 2 parts, and then there's a part 2 where we're testing for the presence of lipids, which I think only has 1 part. So there are 2 parts to the exercise, but 3 different things. I don't know if that clears it up at all. There's 3 kind of different things we're doing. 2 of them are the same. Okay, so now you're going to get your plastic spoon or regular spoon, if that's what you have, and you're just going to scoop a little bit of potato out. You're going to jot down the color of each of the 4 samples and the raw potato into data table 2. And then we are going to make an IKI, or Lugol's iodine pipette. And Again, it needs to be the graduated pipette, not the short stem pipette. So hopefully you remember those are the long ones with the markings on the side. You're going to add 1 drop of solution from the glass bottle into each of the 4 samples and to the raw potato. We're going to observe the color of each of those things. And we've seen a positive test for starch before. Okay, so we're just looking for starch right now. Again, this is very much, until we get to part 2, this is very much a reviews kind of a lab. Okay, it shouldn't be new assays. You should hopefully be reviewing the biological macromolecules right now. And then when you're done, you can pour all of those things down the sink and make sure they get washed out with plenty of water. Check the chat really quick. That's all cleared up. All right. So Wednesdays, actually, let me go back. Before we get to that, is everybody like okay with what we're doing? This should really just be kind of like, okay, I've done some of these things before. Hopefully we have some understanding of what benedicts is. I said bioreds, but what benedicts is and what starch is and what iodine is and how these things are testing for different ones of these biomolecules. But are there questions about stuff? Biomolecules or procedures or okay. So Wednesday's quiz questions. For starters, you should probably know the names, structures, and functions. And again, this is going back to the first exam. You should know the name, structures, and functions of each of those 4. So that's what they look like. I don't have functions here, but let's see. I didn't phrase it as a question, but you should know the functions of each biomolecule. You should know what a dehydration reaction is. And so even if you're not doing the lab until later on this week, like if you're one of those people who wait till Saturday, which is totally fine, please make sure that you at least read through the exploration part of the lab because it covers all of these details. It talks about dehydration reactions. It talks about a peptide bond. Hopefully you know that what benedicts is for, because we've used it in the past. Hopefully you know what iodine is used for and what that test means, because again, we've done that in the past. Sudan 3 is the new test that we're doing, and we're not going to do that until Wednesday, but please check out the lab and make sure that you know what each of these things, excuse me, is actually testing for, is actually looking for. Okay, so if you guys have more questions, I'm going to go ahead and stop recording.